Hi, this is Shane, and I'm going to introduce our third speaker of the session. It's Dr. Christian Guerrero Juarez, who is Chancellor's Advanced Postdoctoral Fellow at UC-Irvine. And the title of his talk is Towards a Multiscale Synthesis of Skin Regeneration, Single-Cell Transcriptomics and Modeling Reveal Functional Fibroblast Heterogeneity in Skin Wounds. Thank you, Christian.
Hi, thank you so much for the opportunity to present my work. Can everyone hear me?
Yes, we can hear you, but we need to see your screen.
Okay, perfect. Yes, so thank you so much for the opportunity to present my work today. My name is Christian Guerrero Juarez, and I'm a postdoc fellow at the NSF Simon Center for Multiscale Cell-Fate Research at UC-Irvine. And today I will talk to you about how we are combining single-cell transcriptomics with mathematical modeling to try to develop a multiscale synthesis of skin regeneration. It is estimated that approximately 100 million patients acquire scars as a result of elective surgeries of trauma in the developed world. Of these, approximately 15% are pathological scars, such as keloids, a type of Wright's scars, as well as hypertrophic scars. Approximately $12 billion is spent annually in the United States alone for scar-related treatments and options. It is also estimated that people with abnormal scarring may face physical, aesthetic, psychological, and social consequences. So in humans, scars usually lack hair follicles and skin fat and often become disfiguring. So what we're trying to do is we're trying to use a multiscale interdisciplinary approach to try to understand and develop skin regeneration therapies to try to shift wound healing towards regeneration and away from scarring. In order to tackle this question, we use the model of wound-induced hair follicle neurogenesis. In this mouse model, we create large excisional skin wounds in the lower dorsum of the mouse, and we let them heal by secondary intention for approximately 1 month. After 1 month, we're able to see that in the center of the skin wound, you are able to regenerate both hair follicles depicted here in the green area, as well as dermal fat. These dermal fat are fat cells that also regenerate and are juxtaposed to the hair follicles that regenerate only in the center of the wound. These two components that regenerate are considered to be the hallmarks of skin regeneration and often are thought to also restore skin function through a regenerative mechanism. Now, how does this mechanism happen? So after the wounds have been inflicted in the mice, the hair follicles begin to regenerate about 2 weeks after the skin has been inflicted. And here I'm showing you an example of what the downgrowth hair follicle forming looks like. After the hair follicle forms, approximately 21 days after the wound has been inflicted, fat cells begin to appear. And in the next few days, they become they enlarge, and they also are able to become lipid-laden fat cells. And these are depicted here. The entire regeneration process takes approximately 1 month. The hair follicles here were labeled with a blue color, and the fat cells are right next to them, and they are stained with a dye that stains fat cells. Now, we decided to focus on fat cells because fat cells are actually very important players and have very important roles in maintaining skin physiology. They play pivotal roles in skin. Not only do they maintain the skin integrity and function and structure, but they also possess metabolic as well as non-metabolic roles. These non-metabolic roles have emerged in the last few years, and they have been shown that these fat cells in the skin are able to talk with hair follicles to maintain and drive their growth. They're also important for healing of small wounds by providing molecules that allow the fibroblasts to begin to migrate and close the wound defect. And they also have been shown to have antimicrobial properties. In fact, when skin is infected with bacteria such as MRSA, dermal adipocytes in the skin expand and begin to express and secrete an antimicrobial peptide called cathelicidin, which prevents the infection by killing and stopping further express of the bacteria. So as you can see, fat cells play very important roles in the skin. And so in order to try to understand how these fat cells regenerate in our model and how we can then be able to use these things that we learned to apply to the clinical setting, we went ahead and performed single-cell RNA sequencing. Now, single-cell RNA sequencing is a very important and powerful technique that allows us to interrogate the heterogeneity of tissues at the single cell level. And the way that we carried out the experiment was as follows. We inflicted large wounds in these mice. We microdissected the skin tissues, and then we isolated single cells from those tissues. And then we processed them through the single cell pipeline, which in a nutshell encases cells as well as barcoded beads into a lipid droplet, and then you're able to capture those cells, perform reverse transcriptome chemistry to be able to identify and capture the mRNAs and convert them into cDNA, and then sequencing them to try to reveal where all the different genes that are expressed in the single cells. Now, when you do that, we were able to capture approximately 20,000 cells from these wounds, and they are shown here in this two-dimensional plot called PISNI. In this PISNI, all the cells that are similarly expressing genes and are therefore similar in their genetic makeup are going to be clustered together. And all of these cells are essentially depicted by individual colors. As you can see, we were able to identify a large number of cells that range from fibroblasts, immune cells, endothelial cells, Schwann cells, as well as lymphatic cells. And all of these cells are essentially the cells that are being present at the wound at this particular time point. So we were interested in the fibroblasts because we know that fibroblasts are essentially the precursor cells of fat cells. So in order to dig this a little bit more, we took those fibroblasts and we subclustered them to try to identify further heterogeneity within those main populations. We were able to identify that there's approximately 12 different fibroblast states that are present in the wound at this particular time point, and those group into essentially three major populations. The PDGFR positive high fibroblasts, which are depicted here, the CRAB-P1 positive fibroblasts, as well as the RGS5 high positive fibroblasts. Now, all of this is great because it's giving us essentially a handle at looking at what types of fibroblasts live in the wounds at this particular time point when prior to the onset of regeneration. But we wanted to see if there's also spatial heterogeneity within these wounds. And so what we did next is we took some of those main fibroblast populations, and then we used antibodies against the genes that are expressed uniquely in these populations to try to resolve their location in the wounds. And so what I'm showing you here is immunostainings against two particular time populations of fibroblasts. One is the CRAB-P1 positive and one is the MES1 positive, both of which are PDG receptor alpha. But an interesting observation is that these two populations are essentially segregated in distinct locations in the wound. One of them are essentially present mainly in the upper dermis of the wound, which in this case is called the papillary dermis. And the other ones are located mainly, segregated mainly to the bottom of the wound, which is called the reticular dermis. So you can see that this single cell RNA sequencing technology allowed us not only to identify all the different types of cells, but if you hone in on the fibroblasts, you're able to resolve a larger degree of granularity in terms of the gene expression profiles and also in the location of where they reside in the wound. And so then we were interested in whether these differences may also be functionally relevant, if have any role in skin regeneration. And so in order to do that, one of the things that we noticed is that a few of these populations express also myeloid markers. And these myeloid markers are essentially markers that are expressed or known to be expressed only in immune cells. And so that was very interesting to us because in the literature, it had been proposed that there is a subset of fibroblasts that are derived from hematopoietic cells or blood cells that have special roles in wound healing and contraction. But nobody has looked at the roles of these cells in the terms of regeneration. And so we were interested in determining whether these cells were in fact doing something else other than grafting into the wound and contracting it. And so we identified that these populations of fibroblasts do exist by doing essentially pseudotime analysis and expression profile with other markers of these putative fibrocytes that people have even identified previously. And we found that these cells indeed are essentially present at the tips of this trajectory, suggesting that it's the end state of the fibroblast differentiation program into myofibroblasts, and therefore could potentially have a myeloid origin. And so then in order to identify this further, we went ahead and validated this observation using distinct methods. The first one was using single-cell Western blot, in which you're essentially measuring protein in single cells. And for this, we used a mouse that genetically labels these cells with cherry protein or red fluorescent protein, and then we stained that also with a marker of these immune cells. And what you can see is that we found essentially the whole range of possibilities. We found cells that don't express anything. We found cells that only express the immune marker, which are purely immune cells. But we also found instances of cells where they express essentially both. The tomato, which is a marker for the fibroblast, and also the lyse, which is the marker of the immune cells. And so that would suggest that these cells are actually present in the wound. And so we went a little bit further, and we saw that essentially these cells are also present in tissue. And you can see here, using the same antibodies, that the cells are colocalizing both markers, suggesting that these cells are present. Furthermore, what we did is we also performed single-cell RNA velocity. And RNA velocity is a high-dimensional vector that takes into consideration the ratio of spliced and unspliced genes to be able to identify or infer the future state of a cell. And so when we did that only on these populations of cells, we're able to see that some of the immune cells actually are showing these vectors pointing towards a fibroblast state, suggesting that this conversion indeed is happening in the wound. And to go a little bit further in validating this notion, we performed bone marrow transplantation assays in which we lethally irradiated mice and then grafted new bone marrow with labeled hematopoietic stem cells using two different constructs. And what we're able to see is that indeed 6 months after this experiment was done, we were able to see that these cells graft in the wound, but not only graft in the wound, they're also able to essentially transform into this period of fibroblasts, and they also essentially make fat cells that are labeled. And lastly, we perform lineage tracing, which is a method to essentially trace the origin of cells using genetic tools. In this case, we labeled immune cells with blue marker, and then we track the origin of those cells in time, and we're able to see that the fat cells that regenerate in the wound are labeled blue, suggesting that they are coming from a cell that once was expressing the lyse marker. And that was done both in tissue as well as in vitro, using two different constructs for them supporting the idea that these cells are indeed coming from a myeloid origin. So then we were able to, we were interested in determining how we are essentially able to model these components using mathematical modeling. And so for that, we developed a two-dimensional hybrid multiscale model in which we model keratinocytes discretely, as well as the fibroblasts and the ECMs, which are components of the wound in a continuum. And so when we model the activators and inhibitors of the fibroblasts, which are the cells that we're interested in the context of wound healing, we are able to see that by modeling not only the wound geometry, or in other words, how severe the wound injury is like, as well as activators and inhibitors in the wound healing process, we're able to see essentially that we get different wound healing outcomes, suggesting that not only that the activator and inhibitor levels present in the wound coming from all these different sources in the wound have an effect on the wound healing outcome, but also the size and type of injury that is induced has a profound effect on that. So we, in this particular model, we only model fibroblasts as one entity, but now with the previous data that we have, we're looking forward to modeling these in the context of regeneration to try to see how heterogeneity within the wound would affect the wound healing outcomes. So taking all of this into perspective, the idea is that we're interested in using all these things that we're learning from our multi-scale model of wound regeneration to try to develop new clinical therapies for skin regeneration. And so just as an example here, we were able to do that at least theoretically in vitro by taking cells from keloids and hypertrophic scars from patients, expanding them in vitro, and then using different methods to try to convert them into fat cells, with the goal of eventually trying to essentially treat patients that have these kind of injuries. So moving forward, I would like to generate a time course of wound healing to interrogate fibroblast, epithelial, and immune cell dynamics a single-cell resolution and use single-cell RNA sequencing data to try to identify the cellular heterogeneity across this wound healing time course to better integrate the pseudotemporal cell heterogeneity aspect of these cells and their gene regulatory networks into this mathematical model to better inform their effects on wound healing and regeneration of skin appendages in mice and eventually to human skin. And so with that, I would like to thank my postdoc advisor, Ching Nie, as well as Maxim Plikas, the rest of the Nie and Plutos lab members, as well as sources of funding. And with that, I would like to take any questions. Thank you.
Thank you so much, Christian.